Hi, Erica.
Oh, hi, Min. Good to see you.
Good to see you. Yeah.
How's things going?
Yeah. Good. Yeah. Right. Yeah. So
Okay.
yeah, it's yeah about 4 p.m. Right at yeah UAE. Right. Yeah.
Yeah.
Yeah. Yeah. So
Well, I will let Le to introduce you and take care of you. I will just be
okay. Great.
listening.
All right.
Thanks.
Okay. Thank you. Yeah.
Okay, maybe just 1 or 2 minutes.
Sure.
Okay, let's just get started. It's my pleasure to introduce Dr. Min Xu for this seminar talk. Dr. Min Xu is an assistant professor at the Computational Biology Department in the School of Computer Science at Carnegie Mellon University. He also served as the training faculty at the joint CNU Pittsburgh PhD program in computational biology. He also served as a training faculty for the Master of Science in Computer Vision program at the Robotics Institute. He is also an investigator at the National Center for Multi-Scale Modeling of Biological Systems. So his research centered mostly around developing computational biology or computational tools to study cellular system using imaging and omics data. He studied his research in the field of computational biology and bioinformatics since 2000, and he has developed many machine learning methods for gene selection, for cancer classification, and cancer gene network discovery and sample phenotype prediction by integrating hundreds of gene expression data sets. Since 2008, he started working in the computation analysis of cellular cryo-electronic tomography data. He designed the structural pattern miming methods and first demonstrated the feasibility of de novo extraction of structures and spatial organization of macromolecule complexes in single cells using cryo-ET data. His current research focus is on developing computer vision and machine learning techniques for cryo-ET derived modeling and the cellular organization at the molecular resolution. So Dr. Shi has published many papers, and including places such as CVPR and MICAI, as well as those prestigious journals and conferences in computational biology, such as PNAS and ISMB. He has received many awards, both from NIH and MSF in United States. He's currently also in the editorial board of statistical method in medical research. Without further ado, I will just hand it the control to Dr. Min Xu. Yeah. Okay. Now the
Okay,
floor is.
thank you. Yeah, okay, thank you very much, Professor Le Song. Okay, and thank you very much also all for inviting me. And it's a great pleasure to be able to present at Mohammed bin Zayed, the University of Artificial Intelligence. So I'm going to give a brief overview of our research on the automatic analysis of cryo-electron tomography using computer vision and machine learning techniques. So as we know, the cell is the basic functional and structural unit of all living organisms. And the understanding of the cell has a fundamental impact to the life sciences fields, such as human health, the origin of life, and environment, food, and energy, etc. So, and if we zoom into a subregion of a cell cytoplasm, we can see that it contains a lot of macromolecules of different structures, there's different spatial organization. And macromolecules are nanomachines that governs cellular processes. So if we want to precisely understand the cellular processes, it is necessary to know the structure and the native structures and the spatial organizations of all macromolecules inside a single cell, and their spatial interactions with other subcellular components. So currently, although we can get a beautiful high-resolution structures of certain macromolecular complexes, there are major limits in the way we obtain such a structure. So first, this structure is obtained using such technique like X-ray crystallography or single particle electron microscopy. And the input of such techniques use many macromolecules with identical structures. And such macromolecules are obtained through a protein purification process. And the protein purification is performed on cell extracts. And these cell extracts are obtained by breaking many cells. So as a consequence of such complicated process, many native molecular structures are destroyed or distorted. Also, once a cell is broken, we lost the spatial organization and the interaction information of such macromolecules. Yeah, so okay, ideally, we want to directly visualize all structures and spatial organizations of all macromolecules together with their subcellular components inside a single cell. However, this information has been extremely difficult to obtain due to the limitations of the data acquisition techniques. Nevertheless, the recent revolution of cryo-electron tomography technique has made such information acquisition possible. So this is an illustration of how this imaging technique works. So first, the sample are quickly frozen and put into a transmission electron microscopy. And then we have these electron beams to pass through this sample to obtain a projection image of the sample. And then we can rotate these samples to obtain a series of projection images. And these projection images are used to construct a 3D image of the cell sample at its close to native state and with submolecular resolution. So therefore, this constructed 3D image contains both native macromolecular structures and spatial organization and their interactions with other subcellular components. And this method become a very promising method to study the structures biology in situ, which means in their original place. And the left figure shows a slice of a 3D tomogram of a subregion of a human cell. The subregion has a volume of approximately 2000 by 2000 by 300 nanometers. The right figures are manual segmentations of subcellular components identified by imaging experts. We can see that such tomograms capture the rich structural features of this subregion at nanometer resolution. However, such structural complexity posed a major challenge in analyzing such 3D images. It would take an experienced cryo-ET expert weeks or even months to carefully inspect the subcellular structures inside a such tomogram. And automation of the identification and segmentation of subcellular structure is critical. And during 2020, the whole world was heavily impacted by the COVID-19 pandemic. And one of the major reasons why this pandemic is very difficult to contain is because the virus is invisible in general sense. On the other hand, under electron microscope, the invisibility does not hold, because in such case, we can clearly see the native structures of the coronavirus particles and many aspects on how they interact with the host cell. Given such data, the native structures of the spike proteins can also be solved together with the other proteins using this computer vision techniques, which can give important insight about the molecular machinery of the infection process. So, the conventional way of localizing macromolecules inside a tomogram is to match the tomogram with the structural templates obtained from other experimental sources. The major limit of such an approach is that it cannot be used to identify the vast majority of unknown structures. Therefore, the usefulness of such template matching approach is very limited. To solve this problem, since 2008, we have been working on developing various methods for structural pattern mining to detect novel and native macromolecular structures inside the tomograms without relying on any external templates. So this is an example of such structural pattern mining analysis result. The left are two slices of two 3D tomograms of two bacterial cells. By applying structural pattern mining methods, we are able to detect tens of different structural patterns, and among which one pattern we found is very interesting. It has a structure that are very well fit with the crystal structure of cryo-ET complex. And there is another pattern we found that fits very well the low resolution structure of ribosome. So in addition, the patterns detected in these single cells also have very distinct spatial distributions as shown in the bottom figure. So the systematic recognition and recovery of such macromolecular structures is very difficult. First, there are significant imaging limits because the tilt angle range is limited. It results in missing data and in the reconstructed tomograms. And also the amount of electron dose sent through the organic material sample has to be very limited. Otherwise, the radiation will damage the molecular structures in the sample. And the low dose results in very noisy tomogram images. In addition, there is a high structural content complexity in such 3D images. So first, a cell can be treated as a very complex living machine. It is substantially more complex than any man-made system, and the native macromolecular structures inside a cell are highly diverse. Secondly, sometimes the macromolecules are inside a very crowded molecular environment in order to perform certain functions efficiently. And thirdly, so far we only know the structures of very small number of macromolecules, and the native structures of the vast majority macromolecules are unknown. And in addition, we have a big data challenge. So each tomogram is a grayscale 3D image in its original size of 6,000 by 6,000 by 1,500 voxels, which is a total of 54 billion voxels. And nowadays, a microscope can easily capture hundreds of such tomograms within a week. And these tomograms could contain even tens of millions of macromolecules of very diverse size and structures. So for the rest of this talk, I'm going to briefly describe the computational techniques we have developed for such structural pattern mining. So these techniques are separated into following categories: subtomogram alignment and averaging, subtomogram classification, subtomogram tomogram segmentation, and high performance computing and software engineering. So the first part is about the subtomogram alignment and averaging for the macromolecular structural recovery. So a subtomogram is a subvolume or a 3D subimage cropped from this big 3D tomogram. And a subtomogram is likely to contain only one macromolecule. So as mentioned before, cryo-electron tomography images are very noisy, and by just directly looking at the raw images, one can generally barely see any macromolecular structures. And a common practice to enhance the structure signal and suppress the noise is to overlay multiple copies of images containing the same structure as shown in figure A. This process is called the image averaging. And subtomogram averaging is a critical step for recovering the underlying macromolecular structures inside subtomograms. However, we have a set of subtomograms. Even if we have a set of subtomograms containing identical structures, we still cannot directly recover such a structure by simply overlaying the subtomograms. This is because despite the structures are identical, they still have different orientations as shown in this figure. And their orientations must be unified before averaging as shown in this figure. And the unification of such orientations is essentially an image alignment process. And in practice, the alignment and averaging is performed iteratively. An intermediate average is formed as a reference, and then the subtomograms are realigned against this reference. And the process iterates until the average does not improve anymore. So, and formally, subtomogram alignment is a process of finding optimal translation and rotation so that the rotated subtomogram can maximally overlap with another subtomogram. However, such alignment is not trivial. Alignment of two 3D images requires the search of a 6-dimensional parameter space, including 3 rotational angles and 3 translations. Such a search is computationally very expensive. And another major effect that will bias such alignment is the missing data in such tomograms. The missing data is due to the limited tilt angle range that can be used for the projection, which introduce orientation-specific distortions in the images. And in Fourier space, such effect can be represented by missing Fourier coefficients in a wedge-shaped region. And this figures shows the orientation-specific distortions are introduced due to the missing wedge effect. And in designing cryo-ET analysis methods, one often needs to take into account of such effects. Ideally, we want to do an exhaustive search over a 6-dimensional parameter space of 3D rotations and 3D translations to find the optimal rigid transformation. However, such exhaustive search-based subtomogram alignment is extremely slow. Only to process several hundreds of subtomograms, one would have to use the computer clusters to spend several weeks. And we have developed, to solve this problem, we have developed a fast alignment method to significantly increase the throughput of the subtomogram analysis. So here I'm going to briefly describe the basic idea of this algorithm. So we first came out a Fourier space representation of the similarity score of two subtomograms as shown in this equation. The score is essentially a Pearson correlation where the denominator has been modified to compensate the missing wedge effect. And we observe that the translation of the second subtomogram only affects the phase term in the numerator. In such case, we can take only the magnitude of the Fourier component to form an approximate score, which is translation invariant as defined in this equation. So, and such approximate score is composed with 3 rotational correlation functions in this equation, and we can then use the spherical harmonics expansion to express each elements in the rotational correlation function as shown in this equation. And then with suitable reparameterization, each of these rotational correlation function can be efficiently sampled over all 3D rotations using fast Fourier transform in spatial coordinate. And finally, we can obtain a small set of candidate optimal rotation angles and corresponding optimal translation and choose the best Regis transformations. And as a result, our algorithm achieved 6,000-fold speed up compared with the conventional exhaustive search-based method. We further propose to integrate our fast alignment method with the maximum likelihood estimation method for improved averaging. This work has been published at ISMB, which is the top conference in the field of computational biology. And since the maximum likelihood-based method use integral of the probability functions to update the parameters, we approximate the exhaustive scanning using the suboptimal transforms obtained by fast alignment as shown in equation 1. Furthermore, we weight each suboptimal transformation using their Van Leeuwen weights in 6-dimensional space. And the computation of Van Leeuwen weights is illustrated in this picture. So basically, each transformation is a vector of length 6 and including 3 translation, 3 rotational parameters. Suppose if we randomly sample uniformly 100 points, then among which 20 points are closest to transformation 1, then the weight for transformation 1 will be 0.2. Yeah. So, and in recent years, the deep learning has significantly advanced the field of image analysis. So besides the above geometry-based subtomogram alignment methods, we recently also developed an unsupervised deep learning and data-driven based subtomogram alignment method. The method was published at CVPR 2020. And given a bunch of pairs of the input subtomograms, our neural network model first extracts the image features. Then it calculates the correlation of these pairs of these image features, and then use it to infer a rigid transformation. The missing wedge effect is also compensated through imputation. The model is trained in such a way that the estimated rigid transformation can maximize the overlap of all pairs of the input subtomograms. And this method is unsupervised method, which means the training data does not contain the ground truth, or we do not need to add the ground truth rigid transformation into the training data. But actually, this model automatically learns the optimal translation and rotation parameters during the training process together with the weights. Okay. The major advantage of such method is this method is a machine learning or data-driven method. Therefore, with more training data available, the model can learn alignment better. And once this model is learned, given 2 testing subtomograms, it can very fastly estimate the optimal rigid transformation. Therefore, it can achieve more than 100 times speed up compared to our previously published geometric-based fast alignment method. So another category of methods that we developed are subtomogram classification. As mentioned before, if all subtomograms contain identical structures, then they can be directly aligned and averaged to recover the structure. However, the macromolecules inside a cell has highly diverse structures. In such case, before averaging, we have to first systematically separate them into subsets of macromolecules of identical structures. This process is called classification. So because we need to process tens of millions of subtomograms, we need to rely on deep learning to process such big data. And this figure gives a basic illustration of how to use convolutional neural network for such classification. Given a subtomogram, a set of learnable convolutional kernels is applied to obtain a set of filtered images. These filtered images are further filtered using another set of convolutional kernels. And the filtered images are further fed to a fully connected layer. And the output of fully connected layer produces a number of binary predictions on whether the subtomograms is assigned to each of the structure classes. Because a subtomogram is a 3D image, we use 3D filters instead of 2D filters that are commonly used in the deep learning-based image analysis tasks. And although the principle of the convolutional neural network is trivial, finding optimal model architecture is an art and requires a lot of explorations. So a common practice in computer vision society is to adopt recent best models for their task of interest. And we follow this approach and adopted two popular network architectures in computer vision domain. So since most of the macromolecular structures are unknown, and also the usefulness of supervised subtomogram classification is very limited. Therefore, rather than using the neural networks for classification, we remove the last softmax layer to extract image features that represents a more continuous likelihood that the input subtomogram being assigned to each class. And we tested whether the supervised structural feature extraction can be used to recover the unknown structures. To do so, we first exclude the subtomograms of one structure class C from the training data, and then use the model to extract the structural features from subtomograms that contain C and other structural classes. This is the t-SNE embedding of the extracted features. So each point is mapped from the convolutional neural network features extracted from a subtomogram in the test data set. And the subtomograms of the unknown structure class C that does not exist in training data are clustered in the red circle region that we can observe. And this indicates that such supervised structural feature extraction can be used for the detection of new macromolecular structures. And we have repeated this procedure by performing a live-out test, and as a result, we are able to successfully recover 16 out of 22 structures. And we further improved the classification models by increasing the number of layers and trying different filters or blocks. And as a result, we found that we have developed 3 new models, and they all outperform the previous models. And we found that this CB3D achieved the best performance. So, and given a trained model that achieve a good performance, in order to further increase the prediction speed to allow processing for even larger number of subtomograms, we also explored the model compression techniques to learn a smaller student model from the big teacher model. Such a smaller student model is able to achieve a substantially faster prediction speed with only a slight decrease of classification accuracy. And as we know, supervised deep learning methods usually require large amount of annotated training data. However, annotating cryo-ET data is very laborious and time-consuming due to the quality controls and the computation cost issues. Therefore, it will be beneficial to have training data from a separate data source where large amount of annotation is readily available or can be obtained in a high throughput fashion, such as using simulation. Then then we can transfer the learned knowledge to a new data set. However, different subtomogram data set have different image intensity distributions due to different experimental conditions. When applying a trained classification model to a new data set with different imaging conditions, the accuracy is usually very low. And this phenomena is called domain shift in cross-data source prediction. To overcome this problem, we adopted an adversarial domain adaptation framework for this transfer learning. The first stage is just normal classification training on the source domain subtomograms. And the second stage is to train a target feature extractor to be domain invariant in an adversarial training fashion. The target feature extractor is trying to fool the domain discriminator so that it could not tell whether the extracted features are from the source domain or the target domain. And in the third stage, since the target domain features extracted are already domain invariant, the classifier obtained from the stage 1 can be directly applied. So experiment shows that our method stably improved the cross-data source prediction as well as outperformed two popular domain adaptation methods. So an alternative approach to domain adaptation, we also developed a domain randomization method, which with the domain randomization, we are able to create a variety of simulated environment with randomized properties and train a model that works across all of them. And likely this model can adapt to the real data, as the real data is expected to be one sample in that rich distribution of training variations. And experiments shows that by combining deep neural network models and domain randomization, we are able to achieve a significant improvement in tasks of classification and thematic segmentation for real cryo-ET data. And comparing to the model trained only on synthetic data that aims to faithfully reproduce the real world data distributions. And we have also developed a semi-supervised learning method to reduce the amount of annotation needed for training. The method makes use of the autoencoder to include a certain amount of self-supervision to help to improve the classification accuracy and in the same time significantly reduce the amount of subtomogram annotation work. The model contains 3 modules. The encoder module extracts features from both labeled and unlabeled data, and the subtomogram reconstruction branch reconstructs the 3D structures from the encoded feature vector, and the subtomogram classification branch takes feature maps output from encoder network and predicts the class label of subtomogram. So extensive experiment demonstrates that our approach can achieve competitive classification performance using substantially less labeled data. It can also improve the reconstruction performance of 3D autoencoder network. Okay. So as I mentioned before, most of the native macromolecular structures are unknown. Therefore, it's difficult to include such unknown structure classes into the training data for our supervised training. So in order to discover new structural classes that does not exist in training data, we have also developed an open set learning method. This method determines whether two test subtomograms belong to the same structure class or not, regardless whether the structure class exists in the training data or not. Specifically, we developed a soft large margin centralized cosine loss function to ensure that the learned features have a large interclass variance and small intraclass variance. Experiment demonstrates that we are able to achieve significantly better verification accuracy and reliability compared to classical loss functions. And few shot learning is another strategy to classify subtomograms whose class do not exist in training data. Different to open set learning that does not require the subtomograms of the new structural class to have any label. Few shot learning consider the case that there exist few labeled subtomograms of the new class. To do so, we first use all labeled subtomograms to train a network to extract the image features and project subtomograms into a low-dimensional space. This is called task agnostic embedding. Okay. And then we train a transformer network to construct a task embedding, which focus on classification of subtomograms of the new classes. Okay, so And at the prediction stage, the query subtomogram X is mapped to both embedding spaces respectively, and then the distance between X and the centers of new class in both embedding spaces are combined as the classification criteria using a nearest neighbor classifier. And our experiment shows that we can classify new structures with a competitive accuracy given only 5 labeled samples of each class. We also adopted a few short domain adaptation method for the learning-based cross-domain subtomogram classification. The essential idea of our method consists of two parts. First, take full advantage of the distribution of the plentiful unlabeled target domain data. And secondly, it's to exploit the correlation between the whole source domain dataset and the few labeled target domain data. And experiments show that our method achieved significant improvement on the cross-domain subtomogram classification. So we also developed an active learning method to guide the annotation process. So we proposed a hybrid active learning framework for querying the subtomograms for labeling from a large unlabeled subtomogram pool. And firstly, our method adopts uncertainty sampling to select the subtomograms that have the most uncertain predictions. To migrate the sampling bias caused by such strategy, a discriminator is introduced to judge if a certain subtomogram is labeled or unlabeled. And subsequently, the model queries the subtomogram that has high probability to be unlabeled. Additionally, our method introduces a subset sampling strategy to improve the diversity of the query set so that the information overlap is decreased between the queried batches and the algorithmic efficiency is improved. So our experiment demonstrates that we can achieve comparable testing performance by using significantly less labeled subtomograms. And the deep convolutional neural networks had achieved great success in image classification. However, convolutional neural networks are vulnerable to specific small perturbations. So we also observed such effect when using convolutional networks for subtomogram classification. So this non-vulnerability of neural networks may pose serious robustness challenges of deep learning models in biomedical image analysis. Therefore, we adopt the state-of-art adversarial attack method to generate adversarial images samples and use such samples to increase the robustness of subtomogram classification. And neural networks have long been criticized for lack of interpretation ability. So it's difficult to know what rules are learned by convolutional neural networks when classifying subtomograms. So we adopted a visualization technique for understanding our trained CNN model. It calculates a weighted combination of feature maps and gradients of the feature maps, and it visualized which regions of subtomogram are important to the determination of its structural class. Such technique is particularly useful for checking whether the convolutional neural network performs the classification using the characteristics structure features instead of some superficially discriminative image features. And this work was published at MBZUAI, which is the top biomedical image analysis conference. And furthermore, to calculate the statistical confidence on whether two macromolecular structures are identical, we also have developed a generative adversarial network-based approach to generate a distribution of randomly sampled pseudo-macromolecular structures. As shown in Figure 2, the generated pseudo structures are visually indistinguishable from real structures. And as shown in Figure 3, we can smoothly interpolate between two structures. This indicates that our method have learned the underlying structural manifold. And in order to further reduce the amount of annotation needed, we also have explored the unsupervised deep learning method to classify subtomograms through clustering. we have constructed a 3D autoencoder and used it to learn how low dimension representation of the input of subtomograms. And then in this low dimensional space, we perform clustering to group these input subtomograms into different structural classes. This is an example on the classified structure patterns embedded to the original tomogram. And we can see that a number of membrane patterns together with the blob-like ribosome, and similar patterns are detected with the distinct spatial distribution. Okay. Another type of work we have been doing is about the segmenting the subtomogram or tomograms. And as mentioned before, the macromolecules could be inside a very crowded macromolecular environment. In such case, if we simply cut out a subtomogram around this macromolecule, then this subtomogram may not only contain the macromolecules of interest, but also some neighbor structures. And the existence of such neighbor structures may bias the recovery and the classification of this macromolecular structure of interest. Therefore, we need to segment out the region occupied by this macromolecule of interest from this subtomogram. Previously, we have developed a number of subtomogram segmentation methods based on geometric heuristics. Later on, we have developed a supervised deep learning method to perform such classification, which achieved and improved the classification performance. The method is essentially a 3D convolutional neural network inspired by fully convolutional network and encoder-decoder architecture. And the one nice feature of deep learning is that it's a high level of flexibility. As an extension, we have also constructed a multitask learning model. The method has multiple branches to simultaneously perform the segmentation and structural classification and the recovery of the core structure. We demonstrated that by simultaneously learning multiple tasks and take advantage of shared image features among tasks, the model can outperform those models that learns each task separately. The tomograms contains not only macromolecules, but also different cellular organelles, such as mitochondria. Therefore, to study interactions of macromolecules with these organelles, it's important to also automatically localize such organelles in the tomograms. To do so, we have also adapted a faster RCM object detection method to automatically localize these mitochondria in these tomograms. And due to the high degree of structural complexity and imaging limits, the automatic segmentation of subcellular component from cryo-ET images is very difficult. It's desirable to develop a generic cell component segmentation method that is first, not specific to particular types of cellular components. Secondly, is able to segment unknown cellular component. And thirdly, fully unsupervised, and it does not rely on availability of training data. As an important step towards this goal, we designed a silence detection method that computes the likelihood that a subregion in a tomogram stands out from the background. Our method consists of 4 steps: supervised over segmentation, then feature extraction and feature matrix decomposition and computation of silencing. The method produces a distribution map that represents the regions of silencing in tomograms. And the experiment shows that our method can successfully label most of silent regions detected by human observer and able to filter out regions not containing the cellular components. So besides the cryo-ET, we have also developed methods for segmenting cells from optical microscope images. And the motivation is to accurately segmenting cells imaged by optical microscopy is an important task in life science study. And in principle, one can use the fluorescence to staining to directly label cells. However, such labeling is not trivial and may introduce phototoxicity to cells. So ideally, we want to directly segment the cells from unlabeled microscope image samples. And so in principle, one could train a convolutional neural network segmenter to perform such a segmentation. However, there are several major challenges. So for bioimages, it's usually hard to get a large amount of training data. And besides, in cell segmentation, there are always less pixels in cell boundary in the microscope images. Another challenge is there are many blurred features in microscope images due to the autofocus and other effects. So taking into account of these challenges, we have developed a hybrid approach for the cell segmentation. And to do so, we first will train encoder-decoder for the convolutional neural network to predict a distance transform instead of predict the segmentation masks. And in the same time, we use the fast RCN model to detect cells and predict the size of the watershed segmentation. And finally, the predicted distance transform and the size are combined to produce the segmented cells. And this method shows high segmentation accuracy on phase contrast and differential interference contrast images. And now I'm going to briefly describe our efforts in software engineering and high-performance computing aspects of cryo-ET analysis. To directly benefit the cryo-ET community, and we are developing an open source AI platform for cryo-ET analysis, incorporating many methods into an integrated system. The analysis system involves methods on both tomogram level and subtomogram level by both traditional geometric-based and state-of-art deep learning-based approaches. So currently, it contains more than 25,000 lines of code, and it has implemented algorithms from 21 papers we published. And as we know, with the advance of the automation of imaging, cryo-ET data is accumulating very fast. So usually a computer cluster is needed for those non-GPU based computational analysis tasks. However, the popular parallel computing tools such as SLORM or MPI are not suitable for completing cryo-ET analysis tasks on a computer cluster shared by multiple users. To solve this problem, we also developed an in-house distributed computation scheduler. So it consists of three components: the analysis programs and the task distribution server program, which manages the execution of tasks generated by the analysis programs and passes the results of each task back to its requesting program. And then workers, which process the tasks in parallel. And many research labs also do not have access to the computer cluster with sufficient computational resources. Also, the hardware and software architectures of computer clusters can vary substantially, and the installation of configuration of specialized software may introduce conflicts with the previously installed software libraries. And therefore, we also provided access to a pre-installed and pre-configured cryo-ET analysis system in the form of a cloud computing service to such labs. And so in summary, we are only at the very beginning of applying AI techniques to the analysis of the cryo-ET data. And we believe that such AI techniques like computer vision and machine learning in particular will play an increasingly important role in facilitating the automatic understanding and analysis of such cellular tomograms. And this work was done by my PhD student Xiangrui Zeng, and together with many other talented students. And our work was also supported by NIH, NISF, CMU, CMLH, and other funding sources. And thank you very much for your attention.
Thank you for the talk. And there's already a question from the audience. Mohammed Yaku asked the question is, how is a subtomogram be chosen? How is it?
Okay. Yeah. Okay, yeah, very good question. Yeah, actually, okay, at this moment, we follow, we use some standard particle picking process, and which does not rely on any templates. So it's called the idea is to use the difference of Gaussian filtering to filter the tomogram, and then select those peaks in the filtered image. And this is a simple method, but currently works okay. But however, there are still many limitations to such method. Therefore, we are trying to also replace such method using other more flexible and more generalized method. Yeah. Such as this silencing detection. It's an attempt to obtain better selection of these subtomograms. Yeah.
Thank you. One question I have is, so your data is like a 3D data, right? And
right.
is a molecule. And how is it different from, for instance, some of the other 3D data we encounter recently, like point cloud? What is the similarity and challenge, unique challenge out there for this type of data?
Okay, yeah. So okay, so this 3D data are the data that on a voxel grid. So that's why it's just a 3D array of the numbers, right? And yeah, that's why it's a 3D grayscale image. And for point cloud, the data is represented as the point coordinates. I would say the tomograms, the cellular tomograms are originally captured as and reconstructed as this 3D grayscale images in the array representation, but one could also convert it into a point cloud representation. In that case, one may be able to recognize those larger structures inside this tomograms. On the other hand, point cloud may not work well when we try to recover those small structures and recognize and recover those small structures, just like those small blobs inside these tomograms, right? And because these images are very noisy, and also this small blobs only the images the subtomograms that containing these small particles are not very large. That's why we may not be able to be accurately represent these small blobs using the point cloud representation. Therefore, probably point cloud representation is not suitable for study these small structures. On the other hand, to study the larger structures and to do the recognition or segmentation of large structures, yeah, that's possible. We could use a point cloud representation to do it, yeah, and which has the advantage of it consumes much less storage.
Yeah, thank you. Kasi, you have a question?
Yeah, so I'm wondering, is there prior information about the cell structure that we can use somehow? All your methods seem to be more like data driven. Is there some
okay.
biological information which we can use to supplement this?
Yeah, sure, of course. Yeah, and yes, and well, Well, I think actually due to the complication of such a image data, there are always, I mean, people in practice are trying to use a combined approach to analyze such a 3D data. And what you can see is only one slice of this 3D image, and imagine how this 3D image, the whole 3D image will look like. And in practice, people will, for example, to use some geometric method or rule-based method to trace out, right, this microtubes or these filaments, and which actually uses the prior knowledge about this structure are supposed to look like a filament or a curve or tubes, right, or other some kind of surface structures. Yeah, these are the prior knowledge. And in terms of other type of prior knowledge, we may able to get, for example, the abundance of a certain type of particles. Maybe that can be also used as a priori knowledge to validate the specific patterns that we obtained. And another type of prior knowledge are this kind of correlated microscope knowledge, which means the same sample was first imaged using the fluorescence microscopy, where this specific protein of interest are labeled, and where in that case we know roughly where this specific protein of interest are located, and then this sample are then imaged using this cryo-ET technique. Then we can, in such case, not visualize the protein of interest, but also bring it to the context together with many other structures. Yeah, so that's why in that case, this labels and fluorescence labels are kind of prior knowledge. Yeah.
So there's one question from the audience. Does the data-driven learning-based method have limitations in 3D shape alignment? And there's a second part. What is the size of data set for training such a model?
Okay, so yeah, the data-driven learning-based method, the limitations, yes, of course. I think this images can be arbitrarily noisy. That's why if the signal to noise ratio is about 0.01, then I think most of the method are fail, just like this data-driven deep learning-based method. Yeah, and yes, and yeah, that's why in that case it's not able to correctly learn the shapes. Yeah, and I mean the alignment of the shapes. Yeah, and also what is the size of data set for training such a model? So yeah, I remember the size is about thousands of subtomograms. Yeah, so yeah.
Thank you.
Sure.
Any other questions from the audience? All right. Yeah, if no more questions, let's thank Dr. Min Xu again, and we'll meet him in the meeting.
Okay. All right. Okay. Okay.
Yeah,
thank you very much for coming. Yeah. All right. Thank you very much for your attention.
Thank you.
All right. Thank you. Yeah. All right. Okay.
Thank you, Mi. Very nice talk.
Okay. Thank you very much,
Nice talk.
Eric. Yeah. Yeah. All right. Okay. Yeah. All right. So I will switch to another link, or shall we? Okay. Yeah. Just one moment. Yeah. So yeah, and yeah.